Hello bachcho, let's see the question. The question is saying that which of the following is correct. Okay, so इस question में हमारे पास तीन statements given है. We have to say which is the correct statement in this question. अगर हम बात करें statement one की, it is saying that autoradiographs are the pictures of resulting DNA sequencing reactions and they are used to enter the nucleotide sequence of a given DNA molecule. So this statement is based on autoradiographs. If we look at the statement B, here we can see that a single-stranded nucleic acid of the known sequence or the origin is used to detect the presence of desired target DNAs and it is known as probe. So here the statement is regarding the probe. Now if we look at the statement C, it is given that the technique of the insertion of a desired gene in the DNA of a plasmid vector is called gene splicing. So it is related to gene splicing. Now if we talk about the key concept that we have to use in this question, then we can say the key concept here is based on different concepts as per the statement. If we look at the statement 1, it is regarding to the autoradiographs. Fine. Let us understand what we mean by autoradiographs. If we talk about this technique, हम कह सकते हैं इस technique में we basically use the X-ray film. हम X-ray film का use करते हैं in order to visualize the molecules. कह सकते हैं we are using it to visualize the DNA fragments and what we are doing here, we can say we are labeling this DNA fragments with the help of radioactive substance. तो हम radioactively इसको label करें DNA को so that it could be seen easily. So in simple terms हम कह सकते हैं in case of radiographs we are using the X-ray film in order to visualize the DNA fragments and here it is giving us the pictures of the X-ray film. Okay, and it is also saying that it is used to enter the nucleotide sequence of a given DNA molecule. Now, another major reason of using this technique is that it can be used to analyze. हम इससे analyze कर सकते हैं the length as well as the number of DNA fragments. Okay, so we can analyze the length and the number of DNA fragments that are separated from one another by using the technique that is gel electrophoresis. So जो statement given है that the autoradiographs are the pictures of resulting DNA sequencing reactions. Correct है because in this autoradiograph we are getting a X-ray based image and also we could separate the different nucleotide sequence of the DNA depending on the length and the number of the DNA fragments. That's why हमारा जो statement 1 होगा that will be the correct one here. चलिए बात करते हैं statement B की. Here the statement B is regarding the probe. Now we have to understand what we mean by a molecular probe. So when we talk about a probe, we can say a probe is a small single-stranded nucleic acid. Either it could be a DNA or it could be a RNA molecule. And this probe, they are showing the complementarity. Okay, उनके पास complementary sequence होती है and they are complementarity sequence it is showing to that of target sequence. तो हमारे पास जो भी टारगेट जीन है या जो भी डिजायर्ड जीन है उसके साथ वो कंप्लीमेंटारिटी शो करता है एंड हम कैसे पता करेंगे कि हाउ द प्रोब इज गेटिंग attached with this target gene because in this case what we are doing हम probe को label कर रहे हैं with the help of radioactive substance. So with the help of radioactive substance we are labeling the probe. So the point at which this probe will go and bind to the target gene it could be seen with the help of a radioactivity. That's why हमें क्या दिया गया है that a single standard nucleic acid of the known sequence or origin is used to detect the presence of desired target DNA is called probe. Absolutely, it is the correct statement here. Fine. Now let us talk about the statement C. If you look at statement C, it is regarding the gene splicing. So अगर हम बात करें gene splicing की, तो gene splicing कैसे हो सकता है with the help of an enzyme known as restriction enzyme. Okay. So in restriction enzyme ko we can say molecular scissors also. Why? Because it will help in dividing the DNA into smaller fragments. So ये किसी भी particular DNA को smaller fragments में break करता है. This is the function of this restriction enzyme. Now let us suppose we have a plasmid vector here. ये हमारे पास plasmid vector है. Now we are allowing this plasmid vector to digest in the presence of restriction enzyme, or we can say restriction endonuclease. Now we know that restriction endonuclease will only bind on that specific part of the DNA that is known as restriction site. तो वो सिर्फ restriction site पे जाके ही bind करेगा, and at that point it will fragmentize the DNA. Okay, so here what we will get a blunted form of the DNA, because ये आर ही यहाँ पे जाके बाइंड कर रहा है, and it is cutting the DNA into two smaller fragments. So we get a blunted form of the DNA. Now we could add a gene of interest. हम यहाँ पे एक gene of interest को add कर सकते हैं with this blunted form of the DNA, and these two are further binded to each other. और इन दोनों को हम फिर से bind कर सकते हैं with each other with the help of an enzyme known as DNA ligase. So with the help of enzyme DNA ligase, we get the recombinant DNA. So hence we get the recombinant DNA, and this recombinant DNA will have the desired properties that we want in any DNA to have. Okay, so हमने यहाँ पे क्या देखा, that the insertion of a desired gene in the DNA of a plasmid vector is called gene splicing. Absolutely correct, because यहाँ पे हमने ये gene of interest लिया और उस gene of interest को हमने insert किया in the plasmid vector with the help of an enzyme, that is restriction enzyme. Again, this is the correct statement. So we have seen that all the three statements here are correct. So we can say the correct option for this question will be simply the option D, that is all of the above statement is correct. So from this we can conclude that the correct answer is option D, that is all of the above statements are correct. I hope you understood the question well. Best of luck.